It's a great question. I'm not aware of any trials in that regard, but it's certainly interesting.
It would be interesting. I think that the challenge with myeloma therapies is that there's frequent administration, so you'd wind up fasting frequently. And one of the issues that we always struggle with at our patients is weight loss. So I think if we're asking our patients to fast for a day before each of their carfilzomib infusions, they're going to get into trouble pretty fast. Yeah, so we usually tell patients, who have multiple myeloma that their immediate family members, in all likelihood won't go on to develop the same thing, but there is an increased risk. When you look at, studies, there's about a 2 to 3-fold increased risk. Now, if I tell you that you're 2 to 3-fold as likely to win the lottery, I wouldn't necessarily advocate that you go out and buy a lottery ticket, because odds are you're going to lose. That said, there are families where there's very strong, linkage, in a variety of different family members, and there is almost certainly either a genetic predisposition or a shared environmental exposure, or both. And there are these genome-wide association studies that have been done. Actually, Gareth Morgan, who's at University of Arkansas now, was previously at MRC in London, was part of the group there. They've actually spearheaded a lot of that work. And there are some, variants, genetic variants in a variety of different genes that might predispose. So, that's always, something to look into. And I know that the IMF at one point, was looking into family predispositions, so it might be worth contacting the IMF to find out where they might be able to go to be tested. But I would certainly have immediate family members, talk to their physicians, depending on their age, about whether or not they should be tested for the presence of an M spike if there's a really strong, family history.
Yeah, I'm not sure which centers still have these open. I know Ola Landgren, who's now at Memorial Sloan Kettering, was doing a study for familial predisposition patients with family members. So they may have a study where you can send blood and they can test. I know that he was doing that when he was down at NIH, and it may still be open there. But the IMF is another good resource. The risk is that as those cells die, they release factors into the bloodstream that can make the patient sick. One is called uric acid, which can cause kidney failure and kidney stones and gout. You can also get release of phosphorus and get all sorts of electrolyte abnormalities. And so as long as you're looking for this, we can generally manage it. Patients get a lot of IV fluids and hydration. They get medicine to bring the uric acid down. And so it's usually not a life-threatening type of situation anymore, as long as we're aware of it and look for it. But that's what can happen.
So it's going to be this year. It'll probably be in the second half of the year as opposed to the first half of the year, but we'll be one of several centers in the U.S. So the Legend Biotech, the Nanjing Legend Biotech company that Adam presented, Janssen has, collaborated with them to basically develop the CAR T product, and we will have that one available at LCI later this year, and we'll be involved in 2 Bluebird cell gene studies later this year as well, but we don't have any that are activated right here and now. Okay, so let me try and tackle this. So not everybody with multiple myeloma, is susceptible to kidney injury when they're first diagnosed. And in large part, it's dependent on the type of antibody, that the myeloma is making. So the most common type of kidney injury with myeloma is something called cast nephropathy or myeloma kidney. And that's typically manifests itself when the burden of clonal light chain antibodies is incredibly high, at least 100 milligrams per deciliter, 1,000 milligrams per liter or higher before you start seeing kidney injury. But every patient's very different. I have patients who have incredibly high levels of serum free light chain levels whose kidney function is completely normal. And then I have other patients where the level is not so high, but their kidneys are clearly affected by the disease. While the disease is active, so if patients have cast nephropathy, and then they get exposed to other kidney injuries, whether it's dehydration because they're sick from their chemotherapy or they've been put on a diuretic, whether they've gotten a CAT scan with IV contrast, whether they've gotten an antibiotic that's also kidney toxic, they are more vulnerable to getting kidney injury. But, the majority of patients with kidney injury from myeloma recover. And once the disease is in remission, and those light chains go back to normal, your risk of kidney injury is no higher than anybody else. I have patients who have bad bone pain, who are now on ibuprofen periodically, and they're doing fine, and their risk of their kidneys being harmed is no higher than someone who doesn't have myeloma. So, but if the disease is diagnosed late and implementation of therapy occurs late, those are the patients that are at high risk of not recovering their renal function. So it's imperative to get diagnosed quickly and get on treatment sooner rather than later.
Yeah, that's the point I was going to make too, I think. Whether it's permanent or not depends on how quickly you can treat it. And if you can get treatment started promptly and get those light chains down, you can reverse it, whereas if it goes on and on for months, sometimes it's not reversible. So it just depends. Yeah, it's a good question, but no, unfortunately, they're different cell populations. So the stem cells you're really trying to get what are called hematopoietic stem cells that are going to give rise to all the blood cells again for a transplant, whereas for the CAR cells, we're collecting more of your T cells, a different immune cell. So they're not interchangeable. You have to do another collection. So not yet, and unfortunately, a lot of the CAR studies that are being run by the companies, if you've had one CAR T, you're excluded from getting another one. But the hope is as we're able to write more trials within cooperative groups or investigator studies, will allow that. There is some effort to try to retreat patients with the same CAR. If you, say, lost your CARs, they lasted a few months, went away, and then the disease relapsed, you could potentially retreat. And that's being done in some of the leukemia studies, and it's just getting started in myeloma as well. I think as early as later this year or early next year. So those trials are already being designed. We have one at Penn that we're trying to open this year.
Is that for newly diagnosed?
It's for high-risk patients who have gotten induction therapy and still have some residual disease. And they're able to get a CAR combination, CART19 and CART-BCMA, if they have very high-risk disease. So there's going to be others like this as well. So for those of you who are unaware, Bill Matsui is a researcher at Hopkins, and he has been proposing for over 20 years that in addition to the majority of plasma cells, there is a subset of population in the bone marrow that express this marker CD19 that looks more like a B cell, a different cell, and that cell is actually responsible for some of the relapse, that it's harder to kill that cell. It's called a myeloma stem cell, and it keeps giving rise to the myeloma over and over again. And this is controversial because other researchers have not always been able to reproduce his findings. And so some people believe that these cells exist and some don't. It's a little bit a matter of faith. And so because we have CART19 cells available to us at Penn, we did a trial where patients got a stem cell transplant and then got CART19 cells, thinking that the high-dose melphalan with the transplant would knock off most of the myeloma cells and the CART19 would come in and mop up the stem cells. And the first patient we treated had a dramatic success. She previously had a prior transplant that only lasted 6 or 9 months and got a second transplant and was in remission for more than a year and a half. But then eventually her disease came back. And so I think the jury's out, to be honest, if these CAR cells are going to work by targeting CD19. Certainly at Penn we are continuing to explore this hypothesis, and that's why that trial I mentioned to you has an arm that's going to get both 19 and BCMA together. And if that looks good, and perhaps showing signs that it might be better than BCMA alone, I think we'll see more trials like that. I think if it doesn't look any different, that's not going to support the hypothesis. And then maybe it won't go forward. So I think it's worth testing, and that's why we're doing it.
I have a question.
Yes.
So is anyone seeing neutralizing antibodies to the CARs with the infusions, such that if you re-challenge, it might not work as well the second time around?
So that was seen in some of the original CAR studies with CART19, particularly in Seattle. They had a murine-based, the CAR was derived from a mouse sequence, and so the patients who got it developed antibodies against that mouse sequence, and when they tried to give it again, it didn't last very long. I don't think it's really been studied well in any of the BCMA yet. The one that we're working with Novartis is a fully human sequence, so it's supposed to have less chance of that. The NCI one was murine, and so I don't know if that will have an impact. It's a good question.